This video series contains scientific and medical concepts aimed at informing healthcare professionals. Please note that the videos may contain visually stimulating images and movement.
Among these detrimental neurophysiological consequences is mounting, though not definitive, evidence for neuroinflammation within ME/CFS, particularly within episodes of post-exertional malaise. Neuroinflammation, in this instance, is defined by microglial cells moving from a resting state to a pro-inflammatory state with associated structural changes as well as functional adaptations, including a local release of pro-inflammatory cytokines such as IL-6, TNF-alpha, and interleukin-1 beta. The presence of these cytokines in specific regions of the brain can lead to a cytokine-induced sickness response, with associated symptoms of heavy fatigue, brain fog, confusion, malaise, and low motivation. In healthy individuals, microglial cytokine release appears to be a transient process meant to occur during, for example, an active infection. Injection of bacterial lipopolysaccharide into the CNS can temporarily induce this neuroinflammatory response, though the microglial cells will rapidly recover to their resting state. In ME/CFS, however, there is mounting evidence supporting the hypothesis that microglial cells are not only easily and abnormally activated, but that these cells appear to be either stuck in this activated position or alternatively are possibly flipping back and forth very rapidly from inactivated to activated. Magnetic resonance spectroscopy is now being used to quantify several metabolites of neuroinflammation within the brains of those with ME/CFS. For example, lactate has been found at levels 4 times greater than in normal controls throughout many locations of the brain, including the ventricles, corpus callosum, occipital cortex, and perhaps most notably within the anterior cingulate cortex, a region known to be intimately tied to the sickness response and to imparting feelings of malaise and depressed mood. This finding is particularly notable because brain lactate only becomes elevated when neurons run out of glucose and when astrocytes begin to produce lactate as an alternate energy source, an extremely rare occurrence usually associated with significant vascular or malignant pathology. Choline, a marker for abnormally high neuronal cell tissue turnover, is also noted to be particularly elevated within the left anterior cingulate cortex of those with ME/CFS as compared to controls. A technique called brain thermometry, designed to assess for brain areas under excessive metabolic demand, has shown that those with ME/CFS have such a ravenous metabolic demand that their brains were measuring, in total, about 1 degree hotter than the brains of controls, with the bulk of this metabolic activity tracking to the very same locations in which lactate and choline were found to be elevated. Positron emission tomography is also being used to measure a radiolabeled isotope ligand to the translocator protein, or TSPO, a microglial cell receptor protein whose expression is magnified when microglia enter their activated cytokine-releasing state. Elevated TSPO levels on PET scans signify activated microglia and have been observed in the thalamus, the cingulate, and the hippocampus, the same regions of the brain found to have elevated lactate among MR spectroscopy studies. Activated microglia were also found within the midbrain and pons of those with ME/CFS, though the implication of this data is unclear. These studies all support the growing hypothesis that microglia appear to be primed or hypersensitized within ME/CFS, and that these microglia may react to very minor triggers in a robust manner. As of 2021, studies are actively underway to discern if large discrepancies within these same metabolite markers exist between rested ME/CFS individuals and those actively experiencing post-exertional malaise.
Not only have localized brain areas shown evidence of neuroinflammation and abnormal function, we are beginning to recognize disruptions to usual healthy patterns of neurobiological activation networks, such as the default mode network in the brains of those with ME/CFS. The default mode network is a collection of brain regions that simultaneously activate in a coordinated fashion when the brain has no external task to perform and is thought to be involved in self-referential thought, mind wandering, social awareness, affective processing, and goal-directed behavior. These regions become deactivated and quiescent when the brain switches from rest to the performance of a cognitive task, activating other brain regions associated with the performance of that task.
The default mode network can be evaluated using imaging techniques such as blood oxygenation level dependent scans, or BOLD scans. Evaluation of the default mode network in those with ME/CFS has shown lower resting neurological activity in all posterior nodes of this network, both before and after exercise. However, strikingly elevated neuronal activity has been detected in the medial prefrontal cortex of the brains of those with ME/CFS, as compared to before exercise, whereas this brain region would show decrement in neurological activity after exercise in healthy controls. The medial prefrontal cortex is actually the primary anterior node of the default mode network, and its abnormal activation after exercise appears to represent an uncoupling of this node from the rest of its neuronal network. It is believed that this uncoupling may be a pathological consequence of exercise in those with ME/CFS, and some have even proposed this clinical feature to be used as a biomarker for ME/CFS.
This video was made possible by the OMF-funded Medical Education Resource Center at BHC, our generous donors, and viewers like you. Please consider making a tax-deductible donation to help keep educational resources like this one publicly available.